Right, so let's start talking about gel electrophoresis. Um, so basically, this is a biotech method for separating DNA molecules by size and charge. And basically, let's talk about how it works. So, say we had a bunch of wells here. Um, there's a plate, and there's a negative end, and there's a positive end. Along with that, you have DNA, which you want to compare for two organisms potentially. So you had like one organism and two organisms, and you want to just see if they're related in some way, potentially a mother and a daughter. So you have two organisms with some DNA and some other DNA. The first thing you need to do is make sure the DNA pieces are pretty small. So one of the things that restriction enzymes can do is they actually cut up the DNA. And when you use the same types of restriction enzymes, they will have the same identification points, which will make the DNA more comparable with each other. So you can say you want to check, you can figure out the differences within the sequence here. In sequence. So say you load up the DNA now, you have the trays and the wells. Which way are they going to start moving? They're going to start moving to the positive charged end. Depending on how the restriction enzymes cut up the DNA, longer ends will always be closer to the negative side, and shorter ends will be towards the positive side. So eventually you'll get something which maybe looks something like this. So you can see somewhat of a relation between the two organisms. One other thing you can use is a DNA ladder, which can be helpful for identifying the base pairs and how large other fragments are in relation to it. So basically, this is like almost a ruler. So say we had it'll give you the how many base pairs long. So let's say this is 200 base pairs. This would be 500 base pairs. And it would increase from there. And you can see the approximate size of the DNA which you have sampled. So basically, it is separated by size and charge, and it can be sequenced later on and probed to find the location of a specific sequence. Yeah. So that's gel electrophoresis. Hopefully that makes a bit sense. And next we'll be talking about microscopy assay.